Good morning, everyone. My name is Yuan Yi, and I'm a regional sales at Oxford Nanopore Technologies. It gives me great pleasure to welcome you all to this exciting webinar exploring the latest innovations in DNA sequencing. Today, we will discover how ONT delivers new sequencing solutions beyond Sanger and learn how Plasmidsaurus is setting a new gold standard by leveraging smart logistics and cutting-edge bioinformatics to deliver ultra-fast, highly accurate sequencing services. Whether you are in academia, biotech, or pharma, this session is designed to help you accelerate discovery and streamline your research workflows. After the talks, our speakers will be taking questions, so please feel free to send in your questions during the webinar using the Questions tab. We will answer as many as we can during the Q&A session after the talks. So let's get started on today's webinar and let us welcome our first speaker, Vanya Vikasa.
Okay, hi everyone. Thank you for joining today's webinar. So my name is Vanya Vikasa. I'm the Field Application Scientist here at Oxford Nanopore Technologies. Before we hand things over later to Nick from Plasmidsaurus, who will take us through the details of the services they offer, I would like to spend a few minutes to go through the introduction of Oxford Nanopore Technologies, offering our key applications, devices, library preparation, chemistry. So at Oxford Nanopore Technologies, we are bringing you the new generation of single molecule sensing technology. Our multi-omics analysis platform supports genomics and epigenomics applications within a single assay, as well as transcriptomics for bulk cDNA sequencing, single-cell sequencing, and direct RNA sequencing. Currently, we have proteomics and metabolomics applications in development. Our technology has validated globally with more than 14,000 peer-reviewed customer publications to date, and the number is still growing. And wherever you are, whether you are working in the field or in the lab, our platform provides accessible and scalable real-time biological insights. Our vision is to enable the analysis of anything by anyone, anywhere, covering different applications ranging from human genetics, cancer, plants and animals, and microbial genomics. We are committed to serve as a foundation for emerging real-world impact, whether it's in the health sector, industry, environment, biosecurity, and also education. So just for a bit of background, I'll walk you through on what happens during the process of sequencing with Oxford Nanopore Technologies. Firstly, we take our extracted DNA or RNA and prep the DNA or RNA with a motor protein that will feed it through a protein nanopore embedded within a membrane. As the molecule passes through the nanopore protein, we applied an electrical current across the membrane. This will create an ionic flow through the nanopore protein. When the nucleotide passes through the nanopore, that ionic current is disrupted and produces signals that with distinct characteristics, we can detect the changes in real time. And those raw signals will be translated into nucleotide sequences. Our technology does not require PCR amplifications. So PCR amplification is optional. So we are not using any optics and will directly sense native nucleotides molecule. This includes base modifications, for example, in a form of methylation. Now we have seen how nanopore sequencing works, but how does that deliver to real-world benefit? There are three key benefits that I want to highlight here. So firstly, nanopore data will provide richer insights. Because nanopore sequencing can sequence native molecules without amplifications or conversion, we can detect base modification of DNA and RNA. Additionally, our technology can sequence any fragment length from short to ultra long, enables us to capture more types of genetics variation from single nucleotide variants to large structural variants within one assay. Secondly, enabling faster time to results. Nanopore data will be streamed real time, so you don't need to wait until the run finish. You can directly analyze the data while you are sequencing for certain applications. This will be very valuable in an environment where you need a rapid decision making. And thirdly, accessibility and affordability. From small scale to large scale, we have the same technology, starting from the pocket-sized sequencer like Minayan to high-throughput production scale systems like Promethean. Now that we've covered the principle of nanopore sequencing technologies, Let's look at one area where these capabilities can really make a difference: plasmid sequencing. So what are plasmids? As many of you already know, plasmids are typically small circular DNA molecules that exist naturally in bacteria. However, they can also be artificially engineered to become critical tools in genetics and biotechnology labs. Plasmid constructs are the backbone of molecular biology, and they serve as a fundamental input material for many uses, from basic research to industrial applications, gene therapy, vaccine development, genetic engineering, and much more. They have enabled fundamental studies into gene function, regulation, and expression, as well as for large-scale production of protein of interest. Plasmids can also serve as factors for gene manipulations, mRNA vaccines, and various applications that contribute to scientific advancement and industrial processes. So as plasmids constructs are fundamentally very important for the downstream applications and experiments, various quality control or QC must be done to verify the plasmids construct. This is to identify any potential errors or mutations before they use the plasmids for further applications. Most of the times, plasmid verification were using Sanger sequencing as a gold standard. However, in practice, it can miss many critical biological information, especially when it comes to plasmid gene therapy and vaccine vector works. So in these slides, we are going to take a look on a few examples on how this happens. On the top left, we can see overlapping peaks, which could be caused by contamination in our samples. It could be caused by more than one colony or the sample prep being contaminated. On the top right, we see that the sequence being terminated. This could be caused by secondary structure on the plasmids, such as hairpin loops or GC-rich region, where the polymerase enzyme will struggle to pass through it. On the bottom left, we see that the sequence becomes unreadable after a stretch of mononucleotides, which likely happened due to polymerase slip after going through a long stretch of mononucleotides. On the bottom right, the signal becomes mixed downstream, which could be due to polymorphic region, which can cause by random mutation or contamination. So now we know Sanger sequencing has their own limitation, especially when it comes to plasmid sequencing. It could only verify certain fraction of the plasmids. This risks missing many critical information such as unexpected mutations, deletions, and unwanted contaminations. On the other hand, Nanopore sequencing can overcome such limitations. Our workflow can rapidly validate the entire plasmid construct, enabling complete plasmid verification, which could reduce failure risk and speed up the discovery process. So compared to Sanger sequencing, where it generally will only look at the inserts of the plasmids, spanning only a few hundred bases and requires specific primers that bind to template DNA during the sequencing reactions with higher relative cost. With offshore nanopore sequencing, on the other hand, we look at the entire complete plasmid. This includes the backbones. This whole plasmid verification allows thorough assessment of the entire plasmid construct, providing a comprehensive overview of its sequence, structure, and potential modification. So you don't need any primers, and with this long-read-based approach, you can easily resolve repetitive regions, structural variants, dimers, and deletions, which can easily be missed by the other methods. It also works across a wide range of plasmid sizes and construct complexities. We provide a simple end-to-end workflow with rapid turnaround time, which will be very handy in rapid development cycles or as a routine quality checkpoints. Let's take a look at a case study demonstrating the utilization of Oxford Nanopore whole plasmid sequencing workflow. This case showcases the capabilities of Nanopore plasmid sequencing in detecting subclonal variants present at low concentration. This could easily be missed by Sanger sequencing, as it cannot reliably call alleles that are present in less than 50% of the DNA molecules. In this study, 4 vaccine templates prepared from spike proteins and pulled at different concentration. And we can see that Oxford Nanopore sequencing can reliably detect plasmid subvariants at 1% of the pool. Now let's take a closer look at what the plasmid sequencing workflow actually looks like using Oxford Nanopore technologies. from DNA prep all the way to data analysis. Here is the overview of the simple end-to-end plasmid sequencing workflow, which we can divide into 4 stages. Firstly, a standard plasmid preparation, picking colonies, growing them in the culture medium, and extracting the plasmid DNA. Next, the extracted plasmid DNA will be prepped for sequencing using Oxford Nanopore rapid barcoding kit. Then it's time for Nanopore sequencing, and finally, the analysis will be using epitomy workflow clone validation. There are some other end-to-end workflows that can offer potential benefits over Sanger sequencing, not only for plasmid verification. For example, you can sequence the entire recombinant AAV with a fast turnaround time and analysis workflow ready to use. And if you have your own sets of amplicon targets, you can also do amplicon sequencing, which optimize for amplicons ranging from 500 to 5000 BP long. There will be no additional primer required after the amplicon product being generated. And our newest addition is a microbial amplicon sequencing, which enable rapid characterization of full-length 16S and ITF regions from microbial samples. And last but not least, our platform also able to sequence cDNA or direct RNA, which will enable mRNA vaccine QC. So that's an overview of the Nanopore sequencing workflow that could have additional value beyond Sanger. Now let's talk about the devices that make this workflow possible. So this is our devices range, which are unified through the same nanopore chemistry, enabling us to support a wide range of use cases and applications, and also a different throughput needs. As we can see from the slides, you will be able to see our range of devices starting from the personal sequencer, MinION, mid-throughput sequencer, GridION, and also higher throughput devices from MetION, which comes in 3 different flavors from MetION 2 devices, providing accessibility and high yield, while ProMethION 24 provides flexibility, which also capable for large-scale, high-throughput projects. Once you've chosen the right device for your needs, the next step is preparing your DNA for sequencing. So we provide several library prep options, but the 2 most widely used are the ligation sequencing kit and the rapid sequencing kit for DNA sequencing. So ligation sequencing kit is optimized for output, and it is our most popular kit. And rapid sequencing kit is more ideal for use where time to answer is critical. The difference between these 2 are you will have more control of the DNA fragment length when you are using the ligation sequencing kit, where in rapid sequencing kit, the transposase enzyme will randomly cut the DNA before attaching the adapters or barcodes. So that gives you a complete picture from the principles of nanopore sequencing through workflows available beyond Sanger, the devices and library prep options you can choose from. And now I will hand this back to Yuan Yi. Thank you.
Awesome. Okay. Please. I think if you stop sharing, I'll be able to share mine.
Okay.
Perfect. There we go. Okay. Hopefully you can see that okay. Okay, so thank you all for being here. So my name is Nick Scales. I'm a technical sequencing specialist at Plasmidsaurus, and I would like to wish a good morning to everybody watching from the Asia Pacific region. I am speaking to you from California, where it is currently yesterday evening, which is kind of fun. And I'm going to tell you a little bit about why you should sequence everything with Plasmidsaurus. So like all of the best companies, Plasmidsaurus was born in a garage. Our CEO, Mark, was working in cell therapy, and he was trying to sequence the constructs that they used for this paper on the left. And they tried everything over and over and found that the plasmid sequencing services available to him were not delivering what he needed, no matter how hard they tried. And it was failing because there was a hairpin in the sequence. And Sanger sequencing is unable to sequence through these for reasons we will discuss shortly. And then he had the excellent idea to try sequencing it with this shiny new technology from a company called Oxford Nanopore, and it sequenced perfectly. So Mark and Kevin formed Primordium Labs in order to shout this from the rooftops and make it available to other scientists. And you can see on the right, this is what the setup originally looked like, although fortunately it's much bigger now. And at the same time that this was happening at the University of Oregon, two professors, Eric and Jim, founded Snipsaurus for providing sequencing services, and the four of them met, and they realized that they had very similar goals, and so these two companies merged in 2021 to form Plasmidsaurus. And the reason that I mention this is because it's important to our identity that our founders are all scientists, and that Plasmidsaurus as a company is fundamentally tied to filling a critical need for the scientists that we serve. So Plasmidsaurus offers sequencing as a service, and these are our three core values. The first is that we're constantly innovating to help scientists move their science faster. We have a big R&D team who innovate on the sequencing workflows to improve the scope, the speed, and the quality of the sequencing. We have some really smart molecular biologists who change and iterate the protocols in order to get the best results. We also have a lot of bioinformaticians, as we have seen that this is often the biggest roadblock, and they're constantly building and updating pipelines in order to translate sequences into answers to your experimental questions. So we don't just provide you with the raw reads, we provide you with biological insights. Lastly, we're building a global network of labs and a huge logistics network to move your samples as quickly as possible. Our number one objective is to accelerate science and help scientists move their experiments faster. As you can see, we serve scientists worldwide with more than 1,000 drop boxes and 10 local labs, which are indicated here by the green dinosaur icon. This network of local labs means that samples spend less time in transit, and for a majority of labs around the world can be run overnight, getting you your results before you start your experiments the next day. And by the way, if you would like us to add a Dropbox near you, please email us because we would love to add a Dropbox right next to your lab. So DNA is the language of biological systems. And if you want to interface with these systems, then you need to modify DNA and introduce it to them. And as I'm certain you know, the most common way that this is done is using a plasmid. Plasmidsaurus was the first company to bring a nanopore-based plasmid sequencing service to market and has been extremely successful because it is unequivocally the better way to do it. Please allow me to explain. So almost 50 years ago, in 1977, Fred Sanger published his method for sequencing by synthesis, building on the principles of PCR. This, without a doubt, one of the most impactful technological innovations of the 20th century. Fun fact to get a sense of just how long ago that was, 1977 was also the year that the first Star Wars movie was released. So the way that this worked is you would use the restriction sites to cut out your insert, you would amplify that and sequence. And what this allowed you to do was confirm the sequence identity of that insert. As in, you could make sure that you were actually inserting the gene that you thought you were inserting. And this is, of course, an extremely important QC step that can save lots of time downstream, as you can very early on identify issues if there are any. But half a century of data has shown us that the method has some problems. Sanger sequencing relies on DNA polymerase, which is an enzyme with a couple of quirks. For example, it really struggles with hairpins, high GC, and repetitive regions. You can see here that the sequencing reads simply stop, and this is real sequencing data visualized in SnapGene. So on the left, you can see an example of an ITR, and that region is highlighted with a little green box. And then on the right, there is a repeat. The red sequencing reads, these are the alignments to the reference. So hopefully you can see quite clearly that in these regions, the reads cannot align. And this is because the polymerase is unable to do its role. It never evolved to make it through difficult structures. Moreover, so because Sanger sequencing is based on amplification, like I said, you cut it out, you amplify it, you can paper over truncated or missing regions. So as an example, if only 10% of the plasmids in your sample are properly formed, you would do this and the method would correctly amplify the good portion. So your results would show everything looks great, but then downstream, your experiment fails. And so what that means is that what you see as a result of the test doesn't actually reflect the truth of what's really there, as in it's not a very good QC test, which is the entire point of doing it. Conversely, you can see the Nanopore reads just sequence right through. Again, this is real data, those are real reads, and just look how much easier that is. Another advantage, as discussed earlier, is that there is no read length limit. So Sanger reads have a limit of about 1,000 bases, which means that if you'd like to sequence a whole plasmid, you would need to sequence multiple times. And it is, of course, possible to do this. People have done this, like this image here shows. Again, this is real sequencing data, but it requires a quite complicated primer walking strategy. So you have to design primers that overlap just enough so that you don't miss any, and they go across the whole length. And so you need to think about melting temperatures, primer diamonds, primer hairpins, the GC content. And so this takes a lot of time. And not to mention that doing multiple Sanger runs also becomes quite expensive. So by using Plasmidsaurus whole plasmid sequencing, you would never need to design primers again. You could sequence the entirety of the plasmid very easily in a single read. And to be clear, it's not no primers because we're designing them for you. They're simply not needed anymore. Sanger sequencing also doesn't work, as alluded to earlier, if you have multiple plasmids, or even if you have a single plasmid, but the primer you design has got more than one primer binding site. You will see mixed peaks. So again, this is a real Sanger trace. And notice at the top, most of the bases are labeled N, and that's because the software is unable to work out what's going on because it's all of the mixture. So Plasmidsaurus whole plasmid sequencing has got no issues with any of those things. High GC, repeats, hairpins, inversions, rearrangements, it sequences right through them, and that's because there's no polymerase. As you saw earlier, we make use of a modified DNA helicase, which, as you can see here on the right, unzips the DNA and also controls the speed at which it moves through the nanopore. The structure and function of the helicase enzyme means that it gets through tricky DNA secondary structures with ease. There are also, like I said, no read length restrictions. So however long that piece of DNA is, will just continue to make its way through the pore. The current record for the longest read is over 4 megabases, which is almost an entire E. coli genome in a single read. Whereas Sanger is limited to just 1 kb. So people usually sequence just the insert or even a portion of the insert. But instead of doing that, what you can do is get this full picture, and you can confirm that the entire plasmid looks the way that you think it should look, including the backbone. As you saw earlier, the library prep uses a transposase to randomly cut the circular DNA, and then wherever it cuts is where the sequencing adapter gets added. So then all of your reads are full length, starting from a different point. And every day, literally every single day, we hear from customers that they finally understand why their experiments have been failing. So they tried to QC their plasmids with the gold standard, and it missed all of this critical information. So in this example, everything had concatemerized, which is something plasmids want to do. And in this example above, there was a big insertion between the kanamycin resistance gene and the origin of replication. And that was totally missed with Sanger. And because this works at the single molecule level, so that means that every individual molecule of DNA is read separately, a mixed sample is really easy to tell. Dimers, trimers, any other concateners are all easy to tell because you see multiple peaks at exactly 2 times and 3 times the expected length. And actually, this is even better than running on a gel, because when you run on a gel, you usually don't see multiple peaks. And that's because the restriction sites also get copied. So you could pull out 3 inserts that were actually in a single plasmid, and on your gel, you would only see a single band at the correct length. But when we sequence them, you see the truth of what is in there. So you can, and it's very easy to tell the difference. So here you can see quite clearly, you've got concatemer on the left, 2 distinct plasmids in the middle, and then a perfect plasmid on the right. And so in general, this is a much better QC step because you simply cannot see this with Sanger sequencing. So to summarize, you're getting the entire plasmid, not just the insert. The single molecule nature allows you to truly see what is in your sample. You'll finally see all of those dimers, truncations, rearrangements, duplications, all of the fun things that biology loves to do. The sequencing is extremely fast. And best of all, there are no primers. There's no primer design, no primer walking, no worrying about primer dimers. You just send it to us and we snip it open with a transposase and sequence the whole thing. So something we pride ourselves on at Plasmidsaurus is speed, like I said, but another is our world-class bioinformatics. And our goal is to deliver actionable biological insights right to your inbox so that you don't need to do anything except begin your next experiment in the morning. So we want you to start that morning with results. So you'll receive an email and it'll look like this, and you can click it. And you click that link, it's going to take you into the main order page of the website. You will immediately see the overall summary of your sequence plasmids alongside a graphical representation of all the main features. So this has been assembled and this has been annotated, and it's got information about each of the genes, as you can see here. You know, the origin, which promoters, which enhancers does it have, which antibiotic resistance genes, etc. But it gets even better. If you upload your reference, we will align it. So then you'll get information about the mismatches, about insertions, deletions, and actually answer the question of what was in your sample. And importantly, did it match what you expected it to be? After the alignment, we will categorize your plasmids with these 3 symbols: a dinosaur, a chicken, or an egg. The dinosaur, that's a perfect match, means that it was an exact match of the reference that you provided. A chicken means that there were 1 or more mismatches, and the egg represents a likely match, but that has one of the 2 nanopore specific sequencing errors. So we think that it will hatch to be a dinosaur, but you should probably look carefully just to make sure. Okay, so say one of your sequences, you're in the website, and one of your sequences has a mismatch. What you need to do is click on it, and again, this is right on the website, and you'll get this much more detailed information. So it's going to take you down to the portion where you can look in further. And here you can see quite clearly, I hope, that there's an entire missing gene. So underneath in the amino acid prediction at the bottom, you can see that it's totally absent compared to the reference. So if you were to use that plasmid, it would still transform the cells would still gain antibiotic resistance for selection, but you wouldn't have any production of the protein of interest. And so with this tool, you can see right after sequencing that this plasmid is no good and should not be used. Another example might be much smaller. So when you open your results on the website, it immediately flags where the problem is located. So in this example, you can see it's right in the middle of the Cas9 gene. And you can see right away that this is actually a very small deletion. So your first question, presumably, would be, what does this mutation mean in the context of the protein? And you can see that this actually has caused a frame shift. And even worse, this frame shift leads to an early stop codon. Hence, you're going to get a truncated protein. So this is exactly the kind of quality assurance information that can save you so much time downstream with failed experiments, right? Preventing the failed experiments. And remember that this is compared across the entire length. So this allows you to see the potential problems that would just be overlooked with Sanger. There's a lot of, I imagine my backbone looks perfectly fine. Well, here you can actually check. Our newest innovation in the plasmid sequencing space is zero prep. So first we abolished primers, and now we're abolishing the mini prep too, thanks to our R&D teams. So with zero prep, you can add extraction for $5 and still get overnight results. So you send us the bacterial colonies just circled on an agar plate, or more commonly, users prefer to pick the colony themselves and send that to us in water or LB, whichever you prefer. And we'll send you back those actionable biological insights. So it's the exact same analysis we just went over for whole plasmid sequencing. Again, there's no primers, no mini preps. You just drop the colonies in the Dropbox. And this typically means that you can save another day or half day per week, which starts to add up to an awful lot of time if you do this regularly. So here is a schematic of where in your cloning workflow you would use zero prep versus our whole plasmid sequencing service. So most commonly users would pick the colony, like I said, and you can send it to us or send most of it to us and then re-inoculate using the same tip on a plate or in liquid. And then that way, while we're doing the sequencing overnight, your plasmid is simultaneously growing. And then in the morning, you'll get your results. You'll see which was the right one, or if it's had whatever property you were hoping it had, and then continue and not lose any time. And we're finding that more and more of our plasmid users are switching to zero prep because why not save a little more time? Our latest announcement, this is last week, has been generating a lot of excitement. So I wanted to quickly talk about that, which is that Plasmidsaurus now does RNA-seq. So what if you could get insights into the functioning of your cells just as easily as sequencing a plasmid, which, as you've seen, is very easy. So traditional transcriptomic studies are very slow and they're expensive, and they require a large minimum number of samples. But with Plasmidsaurus RNA-seq, you can get transcriptome-wide insights in days rather than weeks, and transform your research with a level of speed and insight that has not been possible before. So we are asking for 100,000 cells in Zymo DNA RNA shield. And this is because then there's no cold chain. So they can be shipped at room temperature, which just makes everything easy for everyone. They can just go in the regular Dropbox. And then we'll do the extraction, the sequencing, and the analysis. So this is going to give you unmatched insight at what we hope is an extremely tempting price per sample, only $50 for academic labs, all with a 3-day turnaround time. And I should note that right now, that's for all the sequencing is done in the US for RNA-seq. So it would be 3 days plus whatever the shipping time is here, a day or two. But one thing to note is that we've tested cells in DNA RNA shield, and they're perfectly fine, even for a whole week, and I think slightly longer too. So that shouldn't be an issue on that side of things. And as well, if there is a lot of demand, we could certainly start doing this in our Singapore lab as well, which would speed things up, obviously. So as always, as you hopefully know by now, with Plasmidsaurus, you're going to get cutting-edge analysis built right into the website. So in this case, it's going to be differential gene expression. So which genes were upregulated in response to my treatment or treatments, and which were downregulated. And so when you're uploading your samples, you can set your groups, and you can choose multiple groups and do pairwise or group things together, see all of the comparisons that you would like. And as you can hopefully see, it's highly interactive. So you can also dive into functional enrichment. So you can track the most affected regulatory pathways in your samples. And you can hopefully see that as you click a set on the right, it's going to show you how the specific genes associated with that pathway responded in the volcano plot in terms of fold change versus significance. And there's a lot of other things already that I don't have time to get into. And we're actually continuing to build this out. So if there's anything that you wish that this could do that it couldn't do, please reach out and let us know. Sometimes, of course, a cost-effective targeted approach is exactly what you need. And we have two options. And I'm very glad we had a quick run over of the different Nanopore library prep kits earlier, because that is exactly the difference between these two products. So we have our standard linear PCR. This uses the same approach as plasmid sequencing. So here you have your transposase, fragments the DNA, and that's where the sequencing adapter is added. And this has many of the same advantages. For example, it's very, very rapid. The downside to this approach in the context of amplicons is the length. So a single cut on a circular plasmid is wonderful, because now you've got a sequencing adapter at one end and then the full plasmid sequence. However, if it was already a linear piece of DNA and you have to cut it somewhere to add the sequencing adapter, it's no longer full length, right? It cannot be full length. And it's pretty straightforward to reassemble. So if you have a single amplicon and you do this approach, it's very, very easy to tell, because you have random breakpoints and you can line them all up and see what it was. But this only works with clonal populations for that reason. If you have multiple molecular species in there, then we have the premium PCR method. And this uses the native barcoding method that you saw before, in which the sequencing adapters are ligated to the very ends of the molecule. So this means that all of the molecules are full length, which is, extremely important if you're trying to separate them, because you've got a mixture, because you'll have then all the different lengths and all the different sequences. And this is in our amplicon bracket, but it's really any double-stranded DNA will work with this. Another increasingly popular product of ours is bacterial whole genome sequencing. And this is where you send us the bacterial cell pellet, and we send back complete closed and annotated bacterial genomes. And we're so fast that our users tell us that it's now possible to compress multiple strain engineering cycles into a single work week. So again, just saving you a lot of time. We also do microbiome sequencing if you're ever looking to profile microbial community composition. So this is currently marker gene based 16S, 18S, and ITS. And we love to do extractions. So if you would prefer to send us your samples, saliva, skin, poop, we will do the extraction, the amplification, the sequencing, and the analysis. We also recently launched our AAV sequencing service, which we think is the best AAV QC test on the market. And this is for many of the same reasons as described above. The single molecule nature shows you what actually is in your tube, and this can avoid so many downstream problems. For example, AAVs are really complicated and weird and wonderful, and they love to form these truncated products. And the real problem, biologically speaking, is that the truncated products actually replicate and package more efficiently than the full-length isoforms. So it's extremely common to have these truncation isoforms in your sample, and it's even more important to know that they're there, especially when it comes to kind of thinking about doses and things like that. Yeast genomes, so this is much like our bacterial genome service. We can return an annotated assembly, and again, we love to do extractions too. And similarly, we have eukaryotes in general. So this is separated from yeast, even though obviously also eukaryotic, just because the genome sizes vary so much. So here you can see the pricing is more explicitly based on the sequencing depth. And it's just because a 200 megabase strawberry genome needs a lot less sequencing depth than a 3 gigabase human genome. So at Plasmidsaurus, we think that you should sequence everything. And if you're looking to sequence something that doesn't fit into the products that I've mentioned, then fear not, for we do custom sequencing. So this can be both Nanopore and Illumina based, and those just come in under our custom products. So the process is a tiny bit more consultative. So please get in touch using the support email at the bottom if you'd like to do that. And our goal is to accelerate science and help scientists move their experiments faster. Important step in speeding all of that up is robust quality assurance steps, because like I've said, catching errors early is what saves loads of time downstream, because errors derail things and they multiply and snowball over time. And as you can see from this quote at the bottom, we can usually tell you exactly what went wrong. But I think more importantly, we can help you catch things before anything goes wrong. So at Plasmidsaurus, we're reducing delays, streamlining processes, and delivering richer data, all of which is designed to help our users achieve scientific breakthroughs faster. So thank you for listening. Please note, I've listed a couple of email addresses here at the bottom. So if you have any questions or follow-ups, please email us at the sales@plasmidsaurus.com, or if you prefer a friendly face, then email the very wonderful and capable Maria, and we can set up a call and dive into things a little bit deeper. I'm happy to take any questions now.
Okay, thank you very much, Nick and Vanya, for the insightful discussion. We will be taking in questions from the audiences right now. We will start with the first question that we received. There's a question from, may I know what is the quality required for samples like NGS? What is the high quality of sample needed? Vanya, would you like to elaborate further on this?
Hello, sorry, I will not turn on my camera. So for the quality required for samples like NGS, yes, high quality of sample is needed. And we have several things that we need to check, like sample air concentration, DNA purity, and on some applications, you might want to do fragment analysis to see the fragment length distribution. And for the sample concentration, it will be depending on the protocol you choose as well. So it depends whether you choose the plasmid sequencing or AAV sequencing and other sequencing protocols. And I've pasted a link for more information. You can click the link and see what are the recommended input DNA and RNA QC from ONT.
Thank you, Vanya. So Nick, what about the requirements for Plasmidsaurus services as well? Do you have specific sample requirements?
We have recommendations on our website. They are very similar to the ones on your website because it's the same technology. But yeah, so you could go on either. We also have everything on the website.
Okay. There's also another question. How can we distinguish between a random error in sequencing data and a rare mutation? Is it based on sequencing depth, or do you use an error correction model?
It's both. So I would say, so sequencing things to at least 30x depth tends to be the depth that people have agreed on is sufficient. It is possible, like if you're looking for a very rare variant, then you might need more depth than that. But in terms of sort of generically looking at, do I think something was a sequencing error or not, then typically the distribution of errors is random enough that you can sort of pile them up. And if you see it 29 times it's an A and a single time it was a T, then you could be quite certain that it truly is an A and that one T was a random sequencing error.
All right, Vanya, do you want to comment? Because I would think that most of our audience are more used to Sanger sequencing as a previous gold standard. Do you have any statement on how ONT plasmid sequencing accuracy now compare to Sanger? Vanya, are you able to answer?
Sorry, my internet was cut for a bit. You're asking about the accuracy of Sanger and Oxford Nanopore, right?
Yes.
So when you sequence using Oxford Nanopore, you will be able to get a very high coverage of your plasmids, and therefore you will be able to reach a very high consensus accuracy. It should be 99.9 or 8%. And for the base calling model that we are using now, the standard is the high accuracy model. So it's very high the accuracy is very high already.
Okay.
Yeah, I would say on our side of things, we've done a lot of plasmid benchmarking, and almost every time we see 0 errors compared to the reference. Because there's the raw read accuracy of nanopore sequencing, but when you're assembling a plasmid, like Vanya said, it's very easy to get 1000x depth in like minutes, right? It happens very, very fast because plasmids are very short. And so, yeah, when we compare our assembled plasmids to references, they almost always have 0 errors against the reference.
Okay, so to the anonymous attendee who posted this question, can we clarify the sequencing accuracy of Nanopore versus NGS in premium PCR sequencing provided by Plasmidsaurus? I hope this answers your question. Okay, we have another question. I guess this is for Nick. You haven't mentioned the price for eukaryote sequencing. Will the price be calculated per sample or per GB output or genome size?
It's by the output, and then there's the fee for barcoding on top of that. But everything, pricing, sorry, I'm going to write as I speak, is transparent and can be found on website. For whole genome eukaryotic sequencing, please see this link. And you'll also see there are information about the sample prep there for the earlier question. So here, I can also send the link to the person that had the question about accuracy. We did a benchmarking study that you might find useful. The vast majority of our assemblies have 0 errors. We actually just launched our new website, so if anybody is interested, it's very pretty. We're very happy with it. That's just plasmidsaurus.com.
All right. Do we have further questions from the audience? If not, I think we can close this webinar. So that brings us to the end of today's webinar. Myself and the team would like to thank you all for joining today. If you have further questions about your projects or getting started with nanopore sequencing, please contact support@nanoporetech.com. And if you would like to find out more about sequencing services offered by Plasmidsaurus, please reach out at sales@plasmidsaurus.com. All right, thank you everyone, and I hope you have a good day. Thank you. Bye-bye.
Thank you. Bye.
Bye.